What better way to waltz into the new year than a paper on the effects of dance and cognitive function by Dr. Victoria Y.Y. Zhu. Victoria or Y.Y., over to you.
Thanks very much, Dr. Gendel, and I love the pun. So I'm YY, PGY5 in geriatric medicine. Thanks for joining so early this morning for a discussion on the effects of dance on cognitive function. So I have no conflicts of interest to disclose. So as a brief background, there is a significant interest and need for establishing evidence-based non-pharmacological interventions to promote cognitive health and to prevent cognitive decline in older adults with and without cognitive impairment. Studies have shown that aerobic exercise, coordinative activities, and muscle strengthening can improve cognitive ability. Dance is quite a unique intervention in that it's a complex multimodal activity that involves several different aspects: physical, cognitive, social, as well as emotional, with the potential to improve both cognitive and physical functions in a synergistic manner. I think the artistic expression aspect may differentiate it from aerobic exercise and may increase people's motivation to participate in these interventions. A recent systematic review of dance on neuroplasticity found that dance may actually stimulate beneficial structural brain changes, such as increased hippocampal volume and improved connectivity between cerebral hemispheres by recruiting different motor, cognitive, and somatosensory areas of the brain. Dance has been studied as a therapeutic intervention for improving cognitive and motor function in those with Parkinson's disease. There have also been evidence to suggest that mind-body exercises, such as Tai Chi, yoga, and dance, have been found to have beneficial effects on cognitive function in both cognitively intact and impaired older adults in the realms of global cognition, cognitive flexibility, working memory, and learning. Two other recent meta-analyses found that dance interventions can improve older adults' global cognition and memory, but not executive function, and improve several domains in those with MCI specifically, including global cognition, attention, immediate and delayed recall, and visual spatial abilities. So the study that I'll be reviewing today is a systematic review and meta-analysis on this topic, published in Age and Aging in December 2020 by some local researchers from McMaster University. So with respect to the study question and design, they included studies with older adults aged 55 and over. There was no exclusion for living arrangement or medical diagnosis. So the intervention was dance, defined as mind-body activity of purposeful rhythmic movement to music. And there was no exclusion on the type or frequency or duration of the intervention. The comparator could be any non-pharmacological control group. So for example, education, walking, Tai Chi, being waitlisted, or no physical activity. And studies with pharmacological agents as comparator were excluded. The outcome of interest was cognitive function, specifically executive function, but all measures of cognitive function were considered. All assessment tools in the studies were classified into one of the DSM-5 neurocognitive domains. So the separately published study protocol from 2018 described the search strategy in detail. So the authors searched major bibliographic databases as well as trial registries and conference abstracts for ongoing and unpublished studies. They also searched reference lists for all relevant studies. There were no language restrictions. So of the 3,997 records they identified, 11 RCTs were included in the final systematic review and 8 in the meta-analysis. The risk of bias was assessed using the Cochrane risk of bias tool, with 8 studies rated as low risk and 3 studies rated as high risk of bias. The certainty of evidence was rated with the grade approach. Now looking at some of the results, the 11 studies included 1,412 older adults aged 60 to 80 plus years. There were 7 studies that included healthy older adults without any cognitive impairment. 4 studies included older adults with MCI, defined by the Peterson criteria. And participants were recruited from different settings. So in seven studies, they were from the community, 2 studies from retirement homes, and from clinic settings in 2 other studies. So with respect to the intervention, you can see there was quite a bit of variation. So the frequency of the dance intervention ranged from 1 to 3 times per week. The intensity target was only specified in 3 studies. The total time was 60 to 120 minutes per week. The duration of intervention ranged from 3 months to 12 months. And the majority of studies used ballroom dance type of intervention, and all included some time for socialization as well. In terms of the comparator, in 4 studies, that was participants who were waitlisted, walking in 2 studies, health education in 3 studies, socialization in 1 study, and aerobic exercises in 1 study. So with respect to the main outcome measures, global cognitive function was assessed with the MMSE in 2 studies and the MoCA in 5 studies. So looking at the MMSE results, the mean difference in MMSE scores from the 2 studies was 1.58, which just exceeds the minimal clinically important difference of 1.4 on the MMSE. The evidence was of moderate certainty and only from studies in older adults with MCI. The pooled results of the 5 studies that used the MoCA to assess global function found a higher mean difference of 1.95. The evidence, however, was of low certainty, and the studies included both healthy older adults and those with MCI. There was a subgroup analysis restricted only to older adults with MCI, and it revealed a greater effect in MoCA scores with a mean difference of 6.10, although low certainty evidence. With respect to executive function, this was assessed by the authors as cognitive flexibility and response inhibition. Cognitive flexibility was assessed with the Trails Making Test B in 8 studies, and response inhibition was evaluated with various different tools, but there was insufficient data to include those studies in the meta-analysis. The 4 studies that used Trails B included both older adults with and without MCI, and it showed no effect in cognitive flexibility scores after the dance intervention. A subgroup analysis, though, of patients with MCI found a reduction of 19 seconds on the Trails B. As for the other cognitive domains, learning and memory was assessed as visual spatial learning and memory, and it was only studied in those with MCI, and they found no difference between the groups. Complex attention was assessed by processing speed, so the Trails Making Test A, and the meta-analysis found no difference as well. There was incomplete data for language, and no studies assessed social cognition and perceptual motor function. So looking at the forest plots here that summarize the results for the primary outcomes of global function at the top here measured by MMSE, and then in the second column, second row with MoCA, as well as executive function measured with Trails B. So overall, it showed that dance is associated with an increase in MMSE and MoCA scores, and a decrease in Trails B time. I'll just highlight that the I-squared was for all 3 of these outcomes greater than 50%. As we can imagine, looking at the variety of interventions, indicating moderate to substantial heterogeneity among the studies. The authors also noted that subgroup analyses did not suggest consistently greater effects of dance interventions in older adults with MCI compared to those without. So in summary, just to conclude on the results, the authors found that dance likely improves global cognitive function, and may have a beneficial effect on executive function for those with MCI based on subgroup analyses. They found no effect on complex attention, learning, and memory, and the effect on language was uncertain, as was the effect on social cognition or perceptual motor function. Further research is certainly needed to determine the optimal dose of dance intervention. So in terms of critical appraisal, looking at the validity, so was the research question clearly stated? Yes, I found the PICO question clear. Were the inclusion criteria for studies appropriate? This was described in detail, and they did include a bit of a younger cutoff for the age group 55, but they only included randomized controlled trials in the review. Does it describe a comprehensive and detailed search for relevant trials? Yes, the search strategy, as described earlier, was comprehensive. Were the individual studies assessed for validity? They used the standard Cochrane risk of bias tool and reported the results. Were the results similar from study to study? So as mentioned, there was moderate to substantial heterogeneity among the studies. The study protocol did indicate that outcomes were analyzed in an aggregate manner using random effects model. And then, are the results clinically important? So again, the global cognitive function was evaluated with MMSE and MoCA. It was mentioned that the MMSE mean difference of 1.58 did exceed the minimal clinically important difference of 1.4 in those with MCI. A potential limitation of using the MMSE as an assessment tool, especially in those without cognitive impairment, would be that it does not capture any differences post-intervention. Do these results apply to our patients? Well, in this particular review, the studies included patients who were younger and healthier than patients we may see. Only a minority had MCI, which is certainly a group of interest to geriatricians, given the potential to reduce the risk of progression to dementia. Overall, though, neuroprotective interventions that promote cognitive health are relevant to all older adults in terms of population health promotion. In terms of the conclusions, so this systematic review and meta-analysis of 11 heterogeneous RCTs found that dance probably improves global cognitive function. This is consistent with some of the previous literature on neuroplasticity and other studies. Given the large heterogeneity among studies with respect to dance interventions and assessment tools, the authors call for standardized outcomes and assessment tools, as well as standardized reporting guidelines for these dance interventions. There are several unanswered questions, and sort of left with more questions than answers almost. What is the optimal frequency, intensity, time, and type of dance intervention? Is there a dose-dependent effect? What is the role of other factors in this sort of multimodal experience, the cultural aspects, music, the setting in terms of partnered dance versus group? How does dance actually compare to other forms of exercise, aerobic exercise, and other mind-body interventions such as Tai Chi? So quite a lot of room for further work. So here are my references, and I thank everyone for listening and your attention.
Thank you very much, YY. A phenomenal presentation as always. Generating comments in the chat box, and there are a few hands up and applause in the participant list. One of our methodologists, Jen Watt, has a question which we will take first, and then we will go to Gary with his hand up. Jen?
Thank you, Dev. Great presentation, YY. Can you go back to your slide where you have the forest plots? I think it might be slide 12. Yes, that's
Yeah.
live. Okay, so I have one point of clarification and then one question / hopefully sort of learning point. So for panel B, where you talked about the MoCA score, you mentioned that it had a you can see there's a mean difference of 1.95, and you said, or can you just sort of explain to me again, because I think you had said that it had a beneficial effect on the MoCA. Is that right?
I think that overall it was associated with like the pooled results were favoring dance. I think looking at this, it does cross the 0 line, so like it wouldn't be a significant effect. And I think it was mentioned that one of the studies in particular, the Lazarus study, had sort of pulled things over to favoring dance, and there were some limitations specifically in that study. But I would welcome your comments.
So that's
exactly
that's exactly what I wanted to just bring up, because and you got it. So in panel A, when we're looking at the MMSE, the authors talk about both a clinically and statistically meaningful effect. And as you pointed out, the mean difference and its 95% confidence interval doesn't cross that null, right? And versus for the MoCA, it does. So, the MoCA result isn't necessarily what we would call statistically significant finding, if you will. And then to the second point, and I really like this slide because I think a picture is worth 1,000 words, and that's especially true here. So you pointed out with the numbers, that there's quite a bit of heterogeneity or between study heterogeneity. And so one thing, though, to point out is, and we sometimes think about this when we're evaluating, a meta-analysis like this, is, okay, so the number says there's a lot of heterogeneity, but does it actually change our conclusions? And so if you look at panel A, for example, you can see there's the I-squared is 79%, so that's pretty high, but both of the studies say there's an effect, and their overall conclusion is that there's an effect. So you could say, well, yes, there's heterogeneity there. And so there are differences between these studies which are worth understanding. And so you raised all of those questions, about, what is the intervention? What are the differences? We need to describe these interventions better. But overall, even though the heterogeneity number is high, it doesn't change our conclusion. So that's good. And you could say the same thing about panel B. As you were saying, that one study is really different. So, why is that other study really different? But, and so their I-squared is really high, but, it doesn't change our conclusion. Most of the studies show there's no effect except for that one study. So just always use the picture to help you understand the heterogeneity.
Thank you. Thank you for those tips.
Thank you. Thank you, Jen. Gary, can you unmute, please?
Thanks for a great presentation, YY. In working with my psychology and neuropsychology colleagues, they have, I guess, educated me to how important active controls are. And when I look at dance, which I assume was done in a social group setting, and then look at many of the controls, which appear not to have been done in a group setting, and in this type of study where social interaction may be a key component to benefit, I would just point out that could be a significant limitation in that they didn't really use active controls that would control for the socialization rather than the dance, specifically the dance.
Yes, exactly. I agree with that. I think only one control in one study was socialization.
Great, thank you. And I'll just summarize a few of the comments in the chat box. Barry asks, is the quality of dancing important? Tongue in cheek, he may have 2 left feet. Carol Cohen, similar to Gary's comment, were there other measures such as mood or social interaction measured? A lot of people enjoy the pleasure of dance, and so was that measured? And Dr. Mehta says, were harms looked at, for example, falls? And lastly, Dr. Norris also comments that she found the intervention particularly effective in individuals who were prior dancers. And the last comment, what is the threshold for high versus low heterogeneity in the I-squared score? And so perhaps YY, I'll allow you to respond to those comments and choose which one you'd like to respond as the final one. We'll have to move on.
Sure, yeah, I don't want to overextend my time. So the I-squared in my readings, greater than 50% would be moderate to substantial heterogeneity. The higher would be the more. And I agree with it potentially being better in prior dancers. I didn't look enough into each study about measures of mood and social interaction, but that was not reported in the systematic review. And I think I would hope that quality of dancing is not as important as the effort and time put in. Thank you.
Great, thank you very much, YY. Great presentation and comments. We'll now move on to Dr. Amy Miles, also PGY5, Geriatric Medicine. A very timely presentation, as some of us may be reflecting on the soreness in their deltoid.
All right, thank you, Dr. Gandel, and good morning, everyone. I'm excited to be speaking today about a clinical trial measuring the safety and efficacy of an mRNA COVID-19 vaccine. I have no conflicts of interest to declare. And today my aim is to review the mechanism of action of mRNA vaccines and consider how aging may affect the vaccine efficacy. We will then critically appraise the article and discuss the implications that the results may have on our clinical practice. So as everyone knows, COVID-19 first emerged in December 2019 when a cluster of patients with pneumonia of unknown cause emerged in Wuhan, China. And it was later declared a global pandemic in March 2020 by the WHO. Coronaviruses are large, enveloped, single-strand RNA viruses found in humans and other mammals. Coronaviruses commonly cause common cold type symptoms, but the SARS coronavirus 2 is the third after SARS and MERS to cause severe disease in humans with global spread. SARS coronavirus 2 has distinctive spike proteins that mediate infection by binding to the angiotensin converting enzyme 2 receptor on host cells and facilitating host cell entry. Once the virus enters a cell, viral RNA is released and uses cell machinery to replicate and assemble more virions. The average incubation period from infection to symptom development is about 5 days, and common symptoms include fever, dry cough, shortness of breath, fatigue, myalgias, nausea, vomiting, weakness, and rhinorrhea. However, COVID-19 can present atypically in older adults with symptoms such as falls or delirium. These slides were prepared with Canadian data that was up to date as of January 22nd, 2021. At that time, there had been over 700,000 cases of COVID-19 in Canada. Last week, adults aged greater than 80 had the second highest age-specific incidence rate, and older adults are at much higher risk of complications following infection. As you can see in the graph on this slide, adults aged greater than 80 years made up the largest proportion of patients hospitalized for COVID-19. Adults over age 80 have also suffered the highest mortality rate following COVID-19 infection. Data published by Statistics Canada recently reported that dementia and Alzheimer's were the most frequent comorbid medical conditions associated with severe COVID course, resulting in death during the first wave of the pandemic. There are many more graphs that I could show you that illustrate how the pandemic has disproportionately affected older and more vulnerable adults. Obviously, COVID is an issue relevant to our patient population. Vaccination for prevention of illness is likely to have a major impact on illness outcomes in the pandemic. And older adults are supposed to be prioritized for vaccination soon after those residing in long-term care. Therefore, it's important for geriatricians to understand some vaccine basics in order to counsel older adults about vaccination. So we'll take a bit of time to talk about vaccines. Vaccines are biological products used to safely induce immune response. To achieve this, the vaccine must contain antigens of the pathogen to be taken up by the passive immune system and presented to specialized adaptive immune cells. The adaptive immune response is mediated by B cells and T cells. Most vaccines confer protection mainly through induction of antibodies against the antigen. But vaccines do also induce a T cell response. It's just not well characterized. The immune system of someone who has been vaccinated can more rapidly and robustly mount an immune response when next exposed to the pathogen. Vaccines are usually developed to prevent clinical manifestations of infection, but some may also prevent against asymptomatic infection, resulting in herd immunity and reduced transmission. For highly transmissible pathogens, such as the measles virus, 95% of the population needs to be vaccinated to prevent outbreaks. Less vaccine coverage may be sufficient for less transmissible pathogens. So back to coronavirus. Because the spike protein on the SARS coronavirus 2 mediates contact between the virus and the host cell to cause infection, one way to prevent infection is to block the interaction between the spike protein and the ACE2 receptor via the production of anti-spike antibodies. Therefore, the spike protein, shown in the images on the bottom left, is the target antigen introduced via vaccination to stimulate anti-spike antibodies. There are several different types of vaccines that you can use to introduce antigens. Live attenuated viral vaccines contain a weakened or inactivated virus as the antigen. And other types of common vaccines in use include protein subunit antigens, which are used in influenza vaccines, and polysaccharide protein conjugates, which are used primarily in bacterial vaccines, such as the pneumococcal and meningococcal vaccines. There are new technologies out there that would include viral vectors, which basically include a recombinant viral sequence inside a viral vector, and the viral vector is used to sort of stimulate the immune system and mimic natural infection. And what we'll be talking about today are nucleic acid vaccines, which consist either of DNA or RNA encoding the target antigen, and they use the host cell's machinery to transcribe the DNA or RNA into the antigen. Nucleic acid vaccines are highly versatile. They're fast to adapt to emerging pathogens, and that's what was needed at the beginning of the COVID-19 pandemic. So several different types of vaccine candidates are in development. So far in Canada, only mRNA vaccines have received approval for use. AstraZeneca's viral vector was approved for use in the UK, but the phase 2 and 3 trials are still ongoing in North America. So mRNA is the genetic sequence that encodes proteins. In this case, the mRNA encodes the SARS coronavirus 2 spike protein as the vaccine antigen. And the COVID mRNA vaccines use a lipid nanoparticle to stabilize the mRNA for delivery into the cell. Once the mRNA enters the host cell, it can use the host cell's translation machinery to synthesize the spike protein. And the mRNA has been specifically engineered to resemble a fully processed mature mRNA that would occur naturally in the cell cytoplasm. mRNA is a non-infectious and non-integrating technology, so there is no potential risk of infection or insertional mutagenesis with this vaccine. mRNA is degraded by the normal cell processes, and technological advances have allowed for rapid identification of viral genomes, as well as replication and processing of these genetic sequences, which is why mRNA vaccines were the more rapidly developed vaccines and the first to be approved. However, there are known limitations to vaccine efficacy in older adults related to immunosenescence. The mechanisms by which immunosenescence operates are not fully understood, but are thought to involve decreased naive lymphocytes, decreased lymphocyte repertoire diversity, leading to increased terminally differentiated lymphocyte populations and altered cytokine production. Other studies have shown that older adults can have a diminished response to vaccines, resulting in lower antibody titers and reduced vaccine efficacy. There are strategies such as using high-dose vaccines and adjuvants that have been used for other vaccines to overcome this diminished response. For example, the high-dose influenza vaccines contain 4 times the amount of antigen than standard dose vaccines and have shown superior immunogenicity and better clinical efficacy against influenza infection in adults older than 65. Another example is the use of vaccine adjuvants to improve herpes zoster vaccine efficacy. With the live attenuated zoster vaccine, clinical trials had demonstrated lower vaccine efficacy in older adults compared to younger adults, and also a diminished response in antibodies over time. But with the use of a different vaccine coupled with a liposomal adjuvant called AS01B, they found improved vaccine efficacy with no difference between age groups, and also a better sustained response 4 years after vaccination. So my question after reading through this was, how does the immunogenicity of the COVID mRNA vaccines perform in older adults? And this was evaluated through the phase 1 clinical trial of 2 of the mRNA vaccine candidates. So I found these graphs very difficult to read, but in essence, the top panel shows that the titers of anti-spike immune globulin that were elicited by the vaccines were actually lower in the group 65 to 85 years of age compared to the younger groups. And in the bottom panel, similarly, the 50% neutralizing titers elicited by the vaccine candidates were also lower in the older age group. At the far right side, you can see sort of this bar in gray, and the conclusion of this paper mentions that while the vaccine titers were lower in older adults, they were still higher than titers of antibodies that were found in convalescent plasma of previously infected populations. So to summarize, antibody titers after vaccination with the COVID-19 mRNA vaccines are lower in older adults compared to younger, but the implications for clinical efficacy of these lower titers were not shown in the phase 1 trial. We need the phase 2 and 3 to make conclusions about clinical efficacy. So that brings us to the study for review today, which reports the clinical endpoints of safety and efficacy of the BNT162b2 mRNA COVID vaccine. It was published in the New England Journal of Medicine in December 2020. This was a global phase 2/3 blinded randomized control trial designed by Pfizer and sponsored by BioNTech. Both industry sponsors were involved in data analysis and publication. The trial itself included participants over age 12, but the results presented in the report only include data from the participants aged greater than 16. Participants were healthy or had stable chronic disease. They were excluded if they'd had previous COVID-19 infection, if they were actively infected with COVID. And they were also excluded if they were receiving any other treatment that was intended to prevent against COVID or had previous COVID-19 vaccination. Participants were further excluded if they were immunocompromised, pregnant, or breastfeeding. Participants were randomized in a 1-to-1 ratio to receive the 30 microgram dose of BNT162b2 or a saline placebo injected intramuscularly into the deltoid. All participants received 2 injections 21 days apart, and both injections were of the same product. The trial included efficacy and safety outcomes. The primary efficacy endpoint was COVID-19 incidence from 7 days after the second vaccine dose in those with no prior COVID-19 infection. They did do a secondary efficacy outcome that considered COVID incidence in the whole population, regardless of history of past infection. The primary safety endpoints included adverse events measured from dose 1 to 1 month after dose 2, severe adverse events from dose 1 up to 6 months after dose 2 was also a primary safety outcome, but at the time of publication, there had only been an average of 2 months of follow-up. Local and systemic reactions 7 days after each dose are the final primary outcomes. For the efficacy endpoint, participants were instructed to self-report acute respiratory illness and then undergo evaluation and NP swab by the study personnel. The study defined confirmed COVID-19 as having a positive nucleic acid test within 4 days of suggestive symptoms. Local and systemic reactions were actively sought in the first 6,000 participants to be randomized by participants recording their symptoms in an e-diary for the first 7 days after each dose. Adverse events were actively elicited at 1 month follow-up, and severe events were collected until 6 months. The trial also included a stopping rule that was intended to flag any evidence of vaccine-enhanced disease. This trial was event-driven. 164 total cases of confirmed COVID were calculated to give 90% power to detect a true vaccine efficacy rate greater than 30% with a high probability. 22,000 participants in each group were required to reach 164 events in the 6 months based on an infection rate of 1.3%, but the protocol identifies that the infection rate was expected to change over time. The efficacy analysis included those without evidence of past infection who received 2 doses as randomized without any protocol deviations. Vaccine efficacy was calculated using the ratio of COVID illness rate in the vaccine group versus the illness rate in the placebo group. The probability that vaccine efficacy was greater than 30% was analyzed using a Bayesian beta binomial model with a pre-specified success boundary of 98.6% probability that the vaccine efficacy was greater than 30%. For the safety analysis, descriptive statistics were provided for each of local reaction, systemic reaction, adverse events, and severe adverse events, with an average follow-up of 2 months. Between July and November 2020, 43,548 participants underwent randomization, but the trial used different subsets of this population to measure different outcomes throughout the analysis. There's a full definition of the denominator values in the supplemental appendix. And of note, the modified intention to treat population was only used in exploratory analysis and not for any of the primary outcomes. Looking at the basic demographic table in table 1, both of the groups do appear similar. Close to 50% of the participants were female, and the majority, more than 80%, were white. The average age of participants was 52, but if you look elsewhere in the paper, you can calculate the proportion of older adults included. So those aged over 65 made up 20% of the trial population, while only 4% were aged greater than 75. The supplemental appendix provided a bit more detailed information about the distribution of comorbidities in this population. Again, there doesn't appear to be any major difference between the vaccine and the placebo groups, but overall the population appears very healthy, with only about 20% of participants having any type of Charleston comorbidity. The most frequent comorbidities were uncomplicated diabetes and COPD, both coming in around 7.8%. Of note, there were very few patients with dementia included, only 7 in the active group and 11 in the placebo. This trial did meet its primary endpoint of primary efficacy endpoint. Of the 36,523 participants without evidence of prior COVID, there were 8 cases of confirmed COVID in the vaccine group and 162 cases in the placebo group. That case split corresponds to 95% efficacy and a 99.99% probability that the vaccine efficacy is greater than 30%. The results were also significant in the larger population of both unexposed and previously exposed participants. Results were also presented broken down by subgroup. The vaccine appears efficacious in participants older than 65, with the confidence interval lower bound greater than 60% vaccine efficacy. The number of events in the over 75 group was very low, resulting in a wide confidence interval. Again, the supplemental appendix includes estimates of vaccine efficacy by comorbidity. Vaccine efficacy among subgroups that were defined by either the presence of a coexisting condition or obesity was generally consistent with the vaccine efficacy that was observed in the overall population. And this study was not powered to definitively assess efficacy by subgroup. Shown here is the cumulative incidence of COVID-19 after the first dose in the modified intention to treat population. While the trial was not designed to show efficacy of a single dose, the curve here appears to separate around 12 days after the first dose. Again, it's underpowered for this outcome, but the estimate of vaccine efficacy after a single dose was between 75 to 86%. Shown here are the safety primary outcomes. And so I've only included it for the groups over age 55. For the local reactions, pain at the site of injection was the most commonly reported, and severe pain was only experienced by less than 1% of the reactogenicity subset. Pain was reported less frequently in older adults than in younger, and the local reactions did not increase after the second dose. Looking at the systemic events, which are the 2 panels on the right side of the screen, the headache and fatigue symptoms were common in both the vaccine and the placebo group, but increased in the vaccine group after the second dose. Adverse event analyses were provided for all enrolled participants, except those with HIV who were analyzed separately. 26% of vaccine recipients experienced an adverse event, compared to only 12% in the placebo group. Few participants in either group had severe adverse reactions, and most of the adverse events reported in the vaccine group overlapped with the symptoms of reactogenicity that I presented in the previous slide. So we'll move on to critical appraisal now, beginning with some questions about validity. So the assignment of participants to treatment group was randomized, and the groups were similar at the trial onset, as we saw in Table 1. The follow-up time of patients was sufficient to meet the event-driven efficacy endpoint as the trial was designed, but the mean follow-up of only 2 months reported here is quite short and could miss important safety outcomes. It also does not give us any information about the duration of protection that's provided by the vaccine. The population that was included in the efficacy analysis was not an intention to treat population, and any deviation from the trial protocol disqualified participants from the efficacy analysis. However, everyone who received at least 1 injection was captured in the safety data. Unfortunately, I don't believe that the study elaborated on the details about the group with protocol deviations. They just reported reasons such as they didn't receive the second dose, unclear why. Participants, investigators, and study coordinators were blinded, but there were some unblinded people involved in this trial, including the people administering the injection. And also some sponsor staff were unblinded in the early phases, which was not further explained other than to allow them to participate in rapid result review. Otherwise, it does appear that the groups were treated equally through the trial. Other factors that could potentially impact on the validity are that this was obviously a very highly anticipated, clearly industry-sponsored study that was written, analyzed, and pretty much involvement of the sponsor from beginning to end. Despite the potential validity limitations, the results are very impressive and they clearly show vaccine efficacy. The reactogenetic side effects are expected and are attributable to immune system activation. And in a time where vulnerable populations are dying with very few effective treatments, this trial and other vaccine trials to come are very important. Just to speak to our specific geriatric patient populations, there were very few older adults, older than 75, included in the trial population. And the trial population was overall, I think, much healthier than our typical geriatric patient population, and especially those who reside in long-term care. In terms of treatment feasibility, it is a feasible treatment, despite the need to, for extreme temperature transportation. There's a lot of political issues in the distribution plans and supply interruptions, but this should be a very, very feasible vaccination. Other limitations to this data include the insensitivity to rare but severe adverse events due to the short follow-up time. And there probably won't be any long-term data available comparing the vaccine to placebo because it's now unethical to withhold an effective vaccine in the placebo group. So all of the ongoing adverse event monitoring is going to occur without a comparator. Finally, there are several important questions that this paper does not answer, including what is the efficacy of a single dose regimen? What is the duration of the vaccine efficacy? And what is the efficacy for preventing asymptomatic infection? Today, we did not get a chance to review the report from Moderna's clinical trial, but if you're interested, it is also published online through the New England Journal of Medicine, also in December 2020. Safety monitoring data for Canada is readily available. And so far, adverse events have been reported for 0.015% of all doses administered, with the total serious adverse events quite infrequent, a rate of 0.004% of all doses administered. And finally, if you're looking for more resources about the vaccine for yourself or to help in assisting to counsel patients or address vaccine hesitancy, I would recommend having a look at the Center for Effective Practice website because they have already developed several resources for how to approach these types of conversations with patients. And with that, here are my references. Thank you all so much for listening. And I think it's time for discussion and questions.
Great. Thank you very much, Amy. Very articulate and clear. Excellent presentation. Gary has his hand up. There's a comment in the chat box, but again, Jen Watt, one of our methods experts, has led the question, and we will go question period, and we'll go to Jen for the first question.
Hi, Amy. So that was a really incredible presentation. It was quite a tour de force in your introduction. Those are really great slides. So thank you for that. One question I had is about a point that you raised that I think really, given this topic and how sort of quick we had to be able to get these results, is really important to clarify and to talk a little bit more about. So. Unfortunately, I don't know the slide number, but you talked about how they looked at, the probability that the vaccine efficacy would be greater than 30%. And so I was just hoping that you could kind of explain a little bit more about how they use that number. And I don't mean it to be kind of a read my mind type question. I mean it more in terms of, I hope not anyway. So if it's not clear, please ask me a clarifying question. But sort of how did they use that information? Sort of at what point in their study, and what did it mean for the ongoing methods that they used?
Yeah, so I think in their initial sort of calculations when they were determining what would be the marker of a successful trial, my understanding is that they were aiming to show, they assumed that the true rate of vaccine efficacy was going to be 60%, but they chose 30% as their marker of whether or not the vaccine was efficacious. And I have to admit that I read through the trial protocol, and it was really difficult to understand exactly why they thought that the vaccine efficacy was going to be 60%. And to clearly answer Jen's question, I didn't find it entirely clear how they came up with these 30 and 60% margins.
Okay, actually, that's really good, and you're right. Some of the details were in the protocol and in the appendix. They weren't in the main paper, and so I think that everything you're saying is really good. And so just to add to what you said though, is what they were doing, ahead of time when they designed the protocol, at least as I understand it, is they knew they were going to need to get results quickly, or certainly as quickly as they could, and so they planned interim analyses, which they had to keep an eye on the safety perspective. And so when they were doing that, then they also did these interim efficacy analyses. And so they planned ahead of time. What they were going to say was, if we reach this threshold, then there's overwhelming evidence that this vaccine works, and so we need to move forward and try to get the vaccine further down the pipeline. And then they also had a threshold for if we get these types of results, this is pretty overwhelming evidence that this vaccine doesn't work, and we're going to stop. And so that's how they used this information, and why as you said, the trial itself was really short, but it was because when they got to that interim analysis, and you can see from the one figure, it's just very, very distinct and clear that the vaccine group was doing so much better, that's why they moved forward, even though we know from other trials there can be problems in doing that, but just given sort of the urgency of the situation, that was why they designed the trial ahead of time that way, and why things have moved forward so quickly. So that's really good. I'm glad that you brought up that point when we were talking about how they designed the study. And the other just interesting point that I'll quickly make, but I won't go into details unless you had a question about it, or we can always talk about it later, is that you use the word probability, and it's actually right. So, oftentimes we say, oh, there's a 95% chance, or there's a 95% probability of this, given these trial results, which is actually often not right. But here, given the way they did their analyses, so these are Bayesian analyses, as you pointed out. And so here, when they're talking about probabilities, it really is probabilities. So it's just they did their analyses in a different statistical framework. Anyway, so great job, Amy. Thank you.
Thank you very much, Jen. Gary, very patient with his hand up. Please unmute.
Thanks for a great presentation, Amy. The residents blow me away with the quality of their presentations, so great job. I'm going to stick with a methodologic question. This study was framed as an efficacy study, and in methods, we distinguish between efficacy and effectiveness type of trials. Are you aware of what the distinction is and what the importance of that might be in terms of our interpretation of what we might expect in the real world?
Yeah, so my understanding of efficacy is in an optimal population under controlled settings, what is the efficacy of the intervention for the trial versus effectiveness, which is more so the real world results of how effective this vaccine is going to be, despite the issues in rollout, not having the full or having longer than 21-day intervals in between vaccine doses, which we're experiencing now. And so I think there are going to be a lot of differences between the efficacy population and the effectiveness on a widespread level. And so that, and I think there's a lot of that going around in the media right now.
Yeah, excellent. That's a perfect answer. And of course, for us and for me, let's say for us working at Baycrest, where we have a long-term care population, you know, how will people who are frail, who have multiple comorbidities, as you alluded to what extent will they be able to respond to the vaccine is a quite, you know, we don't know the answer, but we are focusing our attention on that population and making sure that they're getting the vaccine because of how susceptible they've been to adverse effects and mortality from it, and are keeping our fingers crossed that even though there's a likelihood that they won't achieve 95% effectiveness, that they'll achieve sufficient effectiveness that'll be well worth our efforts to make sure that they get vaccinated. So that's exactly right. That's certainly that wasn't part of an efficacy trial to look at populations that are the ones that we're most responsible for. Thank you.
Thank you very much, Gary. A few comments in the chat box. Karen De Silva asks, any knowledge on how a 2-month follow-up vaccine outcome compares to other non-mRNA vaccine studies? That is, have there been many long-term studies done for vaccines in general?
So in my reading, the longest term vaccine trial that I came across was the Adjuvant Herpes Zoster vaccine, which I presented very briefly in the introduction, just in as example, exactly of that sort of like effectiveness in older populations, where they did follow up the efficacy of this vaccine over 4 years. And in this adjuvanted vaccine trial, the efficacy was maintained above 90% for the 4 years of follow-up. So there definitely are trials for other vaccines out there that are more longer term.
Great, thank you. A lot of congratulations in the chat box on a great presentation. Amanda Guardhouse, good to hear and be with you again, has a link to answering questions in the chat box. And Jill Alston asks, are there any thoughts on the increased protocol deviation in the treatment group? Have the reasons for exclusion for protocol deviation been explained? And that'll be the last question.
So not as far as I could find. Like in the supplemental appendix and the trial publication, they mentioned things like if there was an adverse event, then the person didn't receive the second dose, and then if you don't get both doses, you're excluded from the efficacy analysis. With withdrawals from the trial, no longer eligible. So I think, and I'm just guessing because it's not clearly laid out, but if you got COVID in between your 2 doses, then you'd be excluded by the second dose. But that was one of my thoughts as well, is that it was not clear who the people were who deviated from the protocol and were then excluded.
Great. Thank you very much. Thank you to both our presenters today. Amy